we would have the next speaker, who is Stefan Halbherr from InnoMedica holding in Bern in Switzerland, and he talks about another liposome platform addressing unmet medical needs in oncology and neurology. And it is yours.
Good afternoon from my side. I guess everybody's already quite exhausted, so I hope to refresh you up a little bit with something we've been discovering in the couple of last years. It was quite a rough ride, and we had very interesting findings. And as you can see, the talk will be about liposomal technology. And just as a roadmap about my talk, I'll have a couple of words about the platform itself, why it makes sense, about what products we foresee in the next coming years. I will especially focus on our two lead products, one in oncology, which is a liposome containing doxorubicin, and the other one, which is a very experimental thing, liposome to treat neurodegenerative diseases. And I'll finish with how we want to continue. So you may say liposomes are an old hat, and I always say you're probably right, or liposomes are the most oldest thing. Liposomes are as old as life itself. It means that phospholipid bilayers are just one of the best biocompatible things. Our bodies are full of phospholipid bilayers, and that was essentially the key point when we started this project. Which platform should we use? We clearly decided to probably innovate within the liposomal area, as there's so much you can do. And to my knowledge, it's pretty much the only nano platform that can claim one special attribute, being actually healthy by itself. If you use unsaturated phospholipids containing omega-3, etc., there are quite interesting healthy benefits already described from the carrier itself. So we thought this is a nice way to start, combining healthy stuff with medicine that can potentiate each other. They're non-immunogenic, of course, also industry compliant, and we got a lot of inspiration from that. So I think one of the key arguments that haven't been made yet really clearly is the regulatory part, of course. If you make liposomes, you can take out of a rooster of very large libraries of phospholipids that are available in GMP grade, which essentially reduces the amount of work you have to do in GMP about 60%. So we have actually filed our first clinical trial file, and it was roughly 10,000 pages instead of 30,000 pages, which you would need to type when you make a new molecular entity. Of course, also you can have reduced drug testing, so you have usually single species toxicity assessment and not double species, which greatly accelerate things and spare some investings. Physico-chemically speaking, liposomes in our hands are great because you can make gigantic scales. You can go down to very, very small sizes of approximately 25 nanometers. You can have very high particle densities while you manufacture. You have basically, our newest products are just one-step manufacture. You have one bulk, you manufacture, boom, your product is done. You don't have any complex chemistry, purification steps whatsoever. It's a very green chemistry. Essentially 100% of the stuff you put into the product comes also out at the end of manufacturing. And of course, the biological factors are very interesting. So here, a rough overview of the pipeline of InnoMedica with our two lead products, Talidox and Talineuren. The TA stands for targeted, the LI for liposome, and hence the rest you can imagine is doxorubicin, or Neuren is for neuroregeneration, which is for the neurodegenerative diseases. So coming down to Talidox, I think a couple of key parameters are quite different than what we have usually seen in the last years. It's very small. I will come to that in a second. And the surface modification is only on the surface, not on the inside. So the PEG molecules are actually there where they need to be. And the way we load the drug essentially doesn't deform the particle whatsoever, so they're perfectly spherical. They're 35 nanometers in average, they're round, whereas the comparator product we aim to compete with has quite different attributes. And this matters quite significantly when we look at therapeutic activity. This is a 4T1 metastatic breast cancer model. So we can also treat very early, of course, then you get just a flat line of tumor growth, and we thought like, yeah, usually breast cancer is detected rather too late if you're unfortunate, and we wanted to mimic the unfortunate situation too in a lab. So we let the tumor grow for at least 2 weeks, whereas it's known that 4T1 has already heavily metastasized. And it has been interesting to observe that only our nanoformulations actually can prolong life in that setting and quite significantly impact on the primary tumor growth. So you see the Talitaxel is the InnoMedica liposomal docetaxel, one of our newest products, and the Talidox, which is facing clinical trial in the next weeks. Of course, we have always used the same dosages, and it was quite surprising to me to see that the Abraxane nanoalbumin paclitaxel given at 30 was actually causing lethal side effects, I guess because of a failure of immunological compliance of human serum albumin in a mouse model. But yeah, just so to mention. And what we've done then is also to compare another very realistic setting where usually the primary tumors are within a couple of days surgically removed, which we did so too for the mice. And right after the surgical step, we started intervening with chemotherapy, and we saw that our liposomal formulations essentially totally suppressed tumor regrowth for a very long time. However, at the end, all of the animals in this trial succumbed due to the metastasis. So we could not reduce it to 0 or so. When we look at tumor accumulation, we see that our very small formulations have a tendency to deliver more drug to the tumor. And I think besides that, because it's a very dynamic thing, we haven't done this kind of tumor accumulation studies over time. We were rather interested in whether or not the drug is actually released and the therapeutic effect is here. So when we were looking at the data from the GLP toxicity assessment, it was quite interesting to see that even very, very high dosages of Talidox don't induce any kind of hand-foot syndrome, which was very surprising to me because I thought like it's a pegylated liposome, it circulates very long, a little shorter than the calyx liposome, and we would have expected to have a lot of hand-foot syndrome effects, but there was essentially nothing, even at the greatly increased dosages, which have nothing to do with reality. So summing up, we have a very strong tumor suppression, minimal cardiotoxicity, no hand-foot syndrome, and also no other surprising side effects that would have been noted in this GLP tox assessment, whereas our comparators sort of the one or other point have not resolved. So in terms of regulatory compliance, I think it's noteworthy that the TLD1 was categorized by Swissmedic as a known substance with innovation. Hence, we had a very low hurdle to jump into the clinic with this in terms of, in comparison, what other medicines need to fulfill. So the phase 1 trial for Talidox has gained approval in August 2018, and the first patient in will be in the next coming weeks. We collaborate for the trial with the Swiss Group for Anti-Cancer Research, the SAKK. There are 5 Swiss hospitals involved, and the lead is from medical doctor, PD doc, Mr. Markus Jörger. We aim at roughly 30 patients, and we primarily seek solid tumors, also breast cancer, ovarian cancer, most likely due to the known anthracycline benefits in these cases. Of course, the endpoints are quite classic. So to the next one, which was actually an accident in our lab. So we were doing all these biodistribution studies with learning how liposomes distribute, and actually one liposomal formulation was lethal when it was containing doxorubicin, whereas the empty liposome was perfectly tolerated. I was really frustrated because it really also nicely accumulated in tumors, but what do you want to do with a lethal chemotherapy? So we thought like, okay, maybe the liposome has other very interesting features, and we followed it up. We left the chemotherapy outside, and we thought like, what can we put into the liposome that probably would be a good medicine, and we actually put in a neuroregenerative substance for a very good reason, because actually this liposome can be taken orally. It circulates quite long through the bloodstream and then accumulates in the central nervous system and also peripheral nerves. It contains GM1 ganglioside, which is a very well-known glycolipid. It's heavily abundant on neuronal surfaces, and it sticks very well into a liposomal membrane. And it has been described that it has a very interesting pharmacological features in terms of neuroprotection, stimulation of dendritogenesis. It works against Alzheimer's disease tendentially, or Parkinson's disease. There's also another resume of what has been already described, what GM1 can do. Unfortunately, GM1 is a generic molecule, and without any patent protection, no pharma industry will be able to significantly invest into this molecule, and with the liposomal technology, we resolved this. Most inspiring was this study from a colleague of mine, Mr. Schneider from the United States. He showed that in 2013, he treated many Parkinson patients in a placebo, double-blind Parkinson's trial, and he showed that he could really significantly push back the Parkinson's disease and stabilize it over the entire course of treatment for 2 years approximately, with a slight worsening, but not really. It was placebo-controlled and delayed start, and the standard of care is there as well. So we thought this is definitely worth investigating, and the only problem here was the patients got injected twice a day subcutaneously with 100 milligrams of free ganglioside, which caused severe side effects at the injection site, bruises, skin irritation, skin damage, hematoma, and free GM1 cannot be patented. So we resolved this issue by making it liposomal. You can take it orally. It has essentially 0 side effects up to date, and we of course checked different liposomal formulations, and only one formulation actually had this neurotropic kind of a mechanism. We made biodistribution studies with fluorescently labeled carrier of such a kind. In the right part, you see a movie how it distributes over time. Of course, there's a quite significant clearance by the liver, but this is not further any problem. And interestingly, whether or not GM1 is present on the surface doesn't matter at all. So I stopped believing in surface modifications playing significant roles. At least it's not the single parameter that guides biodistribution, obviously. Size seems to matter, but also phospholipid composition, etc. And in this case, we put a lot of GM1 ganglioside in there, and there is so much glycan present on the surface, and the impact on biodistribution is so minimal that I think there's something to learn here. At the end, we went to models of disease, and we replicated this 3 times. Taken short, we were able to reduce the dosage of GM1 given by a factor of 4, while increasing the therapeutic efficacy in this MPTP model of Parkinson's very significantly. So we believe we're almost ready for clinical translation there. So for Talidox, we are thinking about smart clinical strategies, how we want to dose, if we want to combine Talidox with other chemotherapy or also immunotherapies. I think would be a very ideal case, since the liposome doesn't impact that heavily on the immune system, on the hematological system, and hence the combination of the two might be a very powerful approach. There's a companion diagnostic in our pocket based on X-ray technology, since X-rays are taken up far more quick than any other diagnostic mean and should be working just fine. For Talineuren, we have asked Swissmedic for scientific advice to qualify it as well as known substance with innovation, since GM1 has been tested in men and all the other phospholipids we use have been also heavily tested in human subjects already. So it should be a known substance. So we're designing the GLP tox for now and thinking about clinical trials, taking key opinion leaders on board. And of course, we're always busy in getting more funding, building the clean room in a bigger dimension, and develop a couple of other pipeline projects. So thanks to all the InnoMedica employees that relentlessly work with great sacrifice occasionally on this project, and of course all our other collaborators. And thank you for your attention.
Yeah, thank you very much for this presentation on liposomes. I'm happy that there's always something new that people dig out from liposome technology, but there are questions.
Thank you. This was a very nice presentation. Could you give more insights of how the liposomes pass from the gut to the brain? Do you need to have a cell to transport there, or how do they accumulate?
I don't know that exactly. The most close-up look we've had is we have labeled the liposomes fluorescently. We administered them intravenously or orally or subcutaneously, and then we sliced the brain in many thin slices to make a confocal microscopy and detect the dye. And the brains were just full of fluorescent dye. So
does it no matter if they were given orally or IV, so you have the same
IV is a little better, but I think orally you can just give far bigger, far larger amounts, way more comfortably. So I think, if possible, the liposome itself will be applicable in both ways. It's in an isotonic solution, so there is an option to give it intravenously if necessary, but I think, if possible, we want to go for the oral.
Oh, there are more questions?
If I understand you right, you have fluorescently labeled your liposomes, and when that is given orally, how are you then sure that the fluorescence you see in brain is due to liposomes and not just the fluorophore?
Well, if you give the fluorophore, it doesn't go into the brain. That's the negative control, but I haven't shown here that's true. But yeah.
Yeah, there's one more just behind.
Yes, I also enjoyed your talk very much, so thank you. You mentioned at one point that you had to screen many different liposomes in order to get the right properties that were desired. So could you, now looking back, now that you have found the right one, looking back, could you explain why that was the right one? Is there a way to predict the right one, or is it still an experimental art and you just need to go through all the ones that are registered?
I think it was a little both. Somehow it was a rational design. We looked at what materials are available. That's pretty much the first choice, and we discarded all materials that are not available in large bulks in GMP grade materials. And second is, at the beginning of InnoMedica, we manufactured very complex liposomes where we used linker chemistry to link glycans on top of it. And we had always this kind of a vision that we want to have glycans in the liposome, and then this idea with GM1 popped up. I think we wanted to have glycans on the liposome, but if we link them, they would be still somehow unnatural. And by using natural things that have very well described interactions with all kind of other lectins or whatever, I think we hit the nail on the head, because the biocompliance is given if you use natural products. And I guess this is the argument why monoclonal antibodies are so successful. They just work along the logic of the system, and I think liposomes have the same sort of rationale if done correctly. If you use too many synthetic chemical approaches, you will denature the liposome in a way that it's just a foreign entity again. And if you don't do that, I think the compliance is quite well. if not the best possible so far. And then I guess we just got lucky also.
Thank you.
Some more questions for this presentation, Danny?
How do you manage to pegylate the liposomes only on one side?
It's really easy. That I can tell you, but I think I should, the process is the product kind of stuff. I shouldn't probably speak too much. But if you're really interested, we can sign a confidentiality agreement, and then. But definitely, I'll be happy to explain to you a little more if you want. But maybe just for now, I think I should not. I want to keep my job.
Okay. So I think these were the questions for this presentation. This was the last presentation for this session. This was the last session for this conference. Thanks again, all the presenters from this session and from the ones previously, and also to you that you stayed so much time to listen to us. And now I leave it over to the organizers.